All right. Good afternoon, everyone. Today will be our third biology class. Okay. Today, actually, we'll finish up the biology subject lah for the entire Form 5 syllabus. This is our first class for today. So, okay, let me share my screen. You can see the textbook lah, the e-textbook. Okay. So basically, today we'll cover 3 chapters. If the first one will be about plant organization and also all the tissues lah. It means they are the basics one. Okay. So first thing, you have to know like in plants what tissues are there, okay? What types of tissue? We have previously learned in human that there are a few types of tissue in your Form 4 syllabus. So now in terms of plant, we have two types. We have meristematic and permanent tissue. Meristematic means it's not permanent. It can grow and grow and it helps the plant to grow. It keeps growing and moving. Whereas permanent, that means it's already kekal, permanent. It will not move anymore, okay? So meristematic tissue, actually there is 3 types, but in our syllabus there's only 2 types, apical meristem and lateral meristem. The other one is intercalary meristem, that means in between the plant tissues lah, but that's not in our syllabus, okay? Permanent tissue, there's also 3 types, epidermal, which means like the surface lah, epidermis, all those, okay? Ground tissue, you have parenchyma, collenchyma, and sclerenchyma. Vascular tissue, these two are the main one for transpiration and translocation, xylem and phloem. Okay, so permanent tissue are matured tissue. Okay, they have experienced differentiation. Now, this differentiation is not the MS dydx. Okay, this differentiation means that a new cell, once it's already developed, it will undergo this process called differentiation, so it becomes a specialized cell. Okay. That means it becomes a specific cell. For example, this cell will become maybe epidermal cell. This cell becomes gut cell. This cell evolves to become palisade mesophyll cell, for example. So this is what we call differentiation. Okay. Okay, and then you have a few types of tissue. Okay, so here are the structure you must know lah. All right, you must know the structure for the leaf, root, and also the stem lah. Okay, now let's look at the function. Epidermal tissue, okay, outermost surface, we all know already. So they have cuticle. Okay, later when we drawing the leaf structure also, you can see lah. Okay, they have cuticle, which reduce the loss of water through evaporation. Okay, protects leaf from mechanical injuries and pathogen. And then epidermal cell, some of them will evolve into gut cell because they will control the opening of the stomata, and also some is root hair cell to increase surface area lah. Okay. Foreground tissue, the first one is parenchyma. Parenchyma is the simplest living cell, okay? Remember, they are simplest living cell, okay? Thinner cell wall, that means their cell wall is very thin lah, okay? And then it's always in a turgid state, providing support and maintaining the shape of herbaceous plants, okay? It's also involved in photosynthesis because remember, it's a living cell, okay? So helps in the storage of starch and sugar, involved in gaseous exchange, involved in repair and regeneration of plant tissue. Okay. After that, the parenchyma will grow into collenchyma. So collenchyma is also living cells, but it's already matured. Okay, matured into cells that are flexible. So cell walls made up of pectin and hemicellulose. Okay, and then cell walls are thicker than parenchyma lah. Just now the parenchyma, the cell wall is very thin, but now it grows into collenchyma, it will be thicker. Provides mechanical support and elasticity, okay? Sclerenchyma is the final stage, that means consists of dead cell already, okay? And they are matured already, okay? So it's very thick, provides support, mechanical strength, and also help in transport of water and nutrients, okay? And then the final one is vascular tissue. Vascular tissue is the main thing in the plant system, that means there will always be 2, xylem and phloem. Okay, so later on in the upcoming chapters, we also look at it. So xylem is made out of dead cells without the cytoplasm lah. Okay, cell wall of xylem contains lignin. Okay, consists of xylem vessels that are elongated, hollow, and connected to each other lah. That means that the entire xylem vessel it's hollow. That means it has holes inside, and then it's continuous lah. From the leaf all the way to the root is continuous. Okay. Enables xylem to transport water and mineral salt okay, during transpiration. For phloem is for translocation, companion cell, and also sieve tube element okay. Made of living cells, sieve tube with the presence of cytoplasm. Sieve tube do not have any organelles such as nucleus and ribosome as they decompose at maturity stage. So phloem consists of sieve tube arranged from end to end, forming elongated and continuous tube structure. Later on also we'll see the diagram. Okay, and the phloem transport the sugar or organic material from the site of photosynthesis like leaves to other storage to other parts of a plant or storage organs such as roots, fruits, and tubers. All right. Okay, meristematic tissue and growth. So meristematic tissue means that the tissues that are not permanent like I said just now, it will continue to grow. So we have apical meristem and lateral meristem. The first one is apical meristem means that it's at the shoot and the root It will grow upwards and longer Okay, that's apical. For lateral, that means at the side It will grow wider or those Okay, zone of cell growth. Now, in each meristem, especially the apical meristem, there is 3 zones for cell growth. Okay, we have the zone of cell division, cell elongation, and cell differentiation. Okay, so this is what we call primary growth. Apical meristem is involved in primary growth because it grows longer, it grows taller. Okay, lateral meristem is secondary growth, which is thicker and wider. Okay, so the first zone is always called zone of cell division because you want to produce more cells to grow and grow and grow. That's why it's called zone of cell division. Okay. The second one is zone of cell elongation. Once you have all these new cells ready, you elongate to form a longer structure. And then the final one is differentiation. Like I said just now, differentiation, that means once this cell matured already, they will evolve and grow into specific cells. Okay. And then you have the matured tissue. Now you have to see whether it's shoot or root. Shoot is growing upwards, root is growing downwards. So the cell division, the first stage is always at the bottom or the top, depending on which structure. Okay. So leaf primordium, that means it's the start okay, at the root cap, and then you can look through yourself Basically, it's the same thing Okay, and then at zone of cell differentiation, you see it will form xylem, phloem, cortex, epidermis Okay. Okay, so zone of cell growth. Okay, now we know there's 3 zones already. So what happens in each zone? Cell division, of course, a lot of mitotic division taking place. So you see more new cells. Okay, you see at first the very young cells, their nucleus is very big one, and then they don't have a big vacuole. They only have a small vacuole. Okay, but when you move to the cell of elongation, the process occurs called vacuolation. That means all these small vacuole, okay, water will diffuse into this vacuole by osmosis, and then the small vacuole become very big, and then they all fuse together to form a very large vacuole during vacuolation. So because of a very large pressure, that means it will push the plasma membrane against the cell wall. So that means from a very small cell, you can elongate into a very large cell already Okay. So this is zone of cell elongation, and then the third one is differentiation. That means you will differentiate into other types of cell. Once they have reached their maximum size, they form permanent tissue. Cells change their shape and structure to become specialized cells. Okay. So for example, epidermal cell and leaf differentiate and form gut cell. So you know gut cell, the structure is how. And then epidermal cells in root differentiate and form root hair cell. Okay, if you have any question, please ask. No questions, right? Okay. And then, okay, so just now, as I mentioned before, there are two growths already. So the first one is primary growth, the second one is secondary growth Okay. So primary growth, that means is the length and the growth of the stem upwards, and then also the root downwards Okay, the longer and also how to say deeper Okay, so this involves the apical meristem. Okay, at the shoot tail, leaf primordia and shoot primordia grow to form new leaf. Root cap will become exhausted when they penetrate the soil. So, excuse me. So replaced by meristem cell. Okay, for secondary growth is the weave or it becomes thicker That means involves the lateral meristem. Okay. So lateral meristem later also we can see it consists of vascular cambium and cord cambium Okay. Now let's see the process. Okay. What happens at secondary growth? So you see at first at first you have your okay, let's say this is your structure of the stem lah, okay. So you have this one, your vascular cambium, and okay, like this lah, or this one lah, okay. So the one inside is the xylem, okay. One inside is the xylem. The one outside is called the phloem. Okay. So, and then you have your vascular cambium. Inside is the pith lah, and then this one outside is the cortex. And then you have your like your epidermis lah, or you also your cork cambium like that lah, okay. So this is when it's young. Now after it undergoes one time of secondary growth, what happens is this one inside the vascular cambium, it will divide, so it will form another layer inside and outside, okay. So it becomes okay. How do I draw it? Okay, let me try. So it becomes like, okay, so there's like 4 layers already. So you see a new layer forms already lah. A new layer forms. Okay. So the one just now you see inside here original here, inside here is xylem. Original outside here is phloem. So they push out already lah. So that means the one at the outermost is primary phloem lah. The one inside is primary xylem lah, because you push in and you push out. So in the middle lah, it starts to form. In the middle, it starts to form the secondary xylem. And then the outside is secondary phloem. Okay. So if you undergo a lot of time, it will continue to push in the xylem, push out the phloem. Okay. So from the middle, the vascular cambium, it keeps on divide and divide and divide. So this is called secondary growth lah. That's why it becomes more thicker and wider lah. Okay, can you understand? And then the cambium ring, of course, will shed the layer to produce the cork cambium outside. Okay, and then the epidermis also lah. I hope you all understand, kan? So we continue. Okay, so the same thing also lah for the root is the same thing. You can read through. Okay, you'll form cork cell is the same thing lah. That means you'll form more secondary xylem and phloem. Okay, in monocot, even though most monocots do not undergo secondary growth, some do, such as Dracaena species, aloe vera, and agave. Now, just now we already learned the process of primary growth and secondary growth. Now we need to know the differences and also the necessity lah. Okay, so necessity of primary growth, why that it needs to grow taller and also the root have to grow deeper into the soil, because allows maximum elongation of plants for photosynthesis. Okay, can transport product of photosynthesis from leaf to other parts of plant. Primary xylem transport water and mineral salt. Primary xylem provides support lah. Okay, secondary growth, why it becomes wider and thicker? Because it provides stability lah, mechanical support. All these are xylem, phloem, replace old and damaged xylem and phloem tissues, and also produce stronger and thicker bark lah. All this you can read for yourself lah. So you see, primary growth mainly is to obtain the sunlight and also the water, where secondary growth is because of support lah, mechanical support, thicker and wider. Okay. Okay, now the similarities and differences. Both are also growth lah. That means increase the size permanently. Both occurs in woody plants. Both also involve cell division by mitosis. But the differences is primary growth. Okay, it involves the apical meristem, where secondary growth it involves the lateral meristem lah. Okay. Occurs on the stem and roots in younger regions of plant is primary growth, where secondary growth is occurs when primary growth has ceased on mature stem and roots. That means, like just now, you know, the zone of cell division, all those, when the shoot already grow, the mature tissue, that part only starts to undergo secondary growth. Okay, primary growth first, baru secondary growth. Okay, growth occurs longitudinally, and then growth occurs radially. Increases the length, increases the thickness. Okay, then you see lah, what are the tissue and structure involved. Do not have woody tissue, have woody tissue for secondary growth. Thickness of bark is thin for primary growth because it's still young, whereas the secondary growth is thick already. absence of annual growth ring because it's just growing up, so of course you don't have annual growth ring. But this one, because you see it evolved from the secondary, tertiary, that's why there will be annual growth ring for secondary growth lah. Okay. Okay, economic growth, you this one importance you can read yourself lah. Okay, growth curve. Now we have a few types of growth curve in our syllabus. There will be three lah: annual, biennial, and also perennial. So annual, that means every year it will grow once lah. That means every year from its from its germination stage to its death phase lah. Okay. Okay, so plants can be classified based on their life cycles, which are annual, biennial, and perennial plants. Okay. So annual plants, that means like I said just now, one life cycle for a season or a year lah. Okay. So example is paddy, because you know lah, paddy, if you go to like Sekinchan or maybe Kedah, you see the paddy, the harvest is every once a year, right? You grow and then you harvest the paddy once a year lah, like that. And then same for pumpkin plant and watermelon plant. Okay, biennial, that means there will be, it will take 2 years lah. Okay, the first year is vegetative growth, that means growth of roots, leaves, and stem structure. The second growth season is reproduction lah. Okay, so example is silvercock, scone plant, cabbage, and carrots. Perennial, that means a lot of years, more than 2 years lah. It will just continue to grow. Example is hibiscus lah, and mango and grass. Okay, now look at the growth curve. Actually, it's also very easy to identify and also explain lah. So you see growth curve of annual plants, that means it's one time lah. The growth curve is 1 year only, one time like that only lah. Okay, so the growth curve is a sigmoid curve. Sigmoid means like an S shape like that lah. So the first stage is decreasing dry mass. Decreasing dry mass, we see. I think exam question also objective got asked what is the stage A lah. Okay, that means the dry mass will decrease. Why? Because you see, inside the seed, the cotyledon, they will use the food stored in the cotyledon to use for germination. Because your seed, you cannot conduct photosynthesis to obtain your own food, man. That's why the cotyledon will use the food inside to germinate lah. Okay, and then stage B only, it will increase lah because it's growing and then photosynthesis, everything. Stage C is constant, that means the rate of growth is 0 lah. Okay, and then the next one will be decreasing dry mass lah. That means happens slowly because of aging and then dying already lah. Okay, for biennial plants is the same thing lah. Okay, growth curves have 2 sigmoid curve. Okay, the first one is produce leaf, photosynthesis, all this lah, the vegetative growth. The second one is food stored, is used to produce flowers and seeds and also for reproduction. Perennial growth, the curve is a lot lah. That means can be 10 curves like that. So it form a very nice curve lah upwards. Okay, small sigmoid curves, and then rate of growth is high during spring and summer, high light intensity, and then rate of growth decreases in winter lah. Very easy only lah. Okay, so these are all the growth curve. Okay, so that's all for chapter 1. Just a very basic introduction to organization of plant tissues and growth lah. Okay. Okay, can you all understand before we move to chapter 2, the leaf structure and function? Chapter 2 is more detailed lah. Chapter 1 is just a very basic intro only. Plants normally people, students, they don't struggle for plants lah, because plants is quite general. Okay, so let's look at plants. Okay, so everyone knows what is a leaf, right? So structure of a leaf, okay, we have lamina and petiole. Okay, external structure consists of lamina and petiole. So lamina, okay, so you see now you're a biology student. So whenever you see a leaf, actually if you are a biology student, you shouldn't call it a leaf lah, because leaf is too general. You can call it lamina. You see all those very flat green surface we call leaf, actually it's called lamina lah. So if you go out, then if you are a biology student, you can say lah, okay, this is lamina. Okay, flat, thin, smooth, and green part of the leaf lah. So lamina is flat shaped to provide a wide surface. Okay, and then gaseous exchange lah. Petiole is the stalk lah that supports the branch or the stalk that supports the lamina lah. Okay, so that's for the structure of leaf. Now internal structure, this is very important. Okay, so before we go on, you have to know that all these lamina and leaves, they are arranged in a pattern called leaf mosaic. That means you see Like this is the plant. Okay, this is the plant. Okay, let's say there's a leaf here. Okay, there's a lamina here. There won't be another leaf that is at the same position on top that will cover it. No, the leaf will maybe move towards here. There will be a lamina over here. Then maybe like another structure over here like that. You understand what I mean? That means there will be different arrangement lah. Okay, it won't cover each other. This leaf on top won't exactly cover 100% of the leaf bottom. It's arranged like variously so that it produce a leaf mosaic lah, in order to absorb maximum sunlight for photosynthesis lah. Okay, now I look at the structure of leaf. I think everyone also knows already this one. So you have the cuticle on the which is the outer waxy surface lah, waterproof and must be transparent. Remember, the epidermis and cuticle must be transparent. If you are not transparent, the sunlight cannot enter the leaf for photosynthesis. Okay, so cuticle prevents the loss of water through evaporation and then also allows sunlight to pass through it lah. Okay, upper epidermis, also upper surface of the leaf, and must be transparent. Cannot have chloroplast. If the upper epidermis already block the sunlight, conduct photosynthesis, then the one inside cannot conduct, right? So that's why it have to be transparent, cannot conduct photosynthesis, no chloroplast. Okay, lower epidermis also same thing, but the special thing about lower epidermis is because there will be gut cells. Okay, so the gut cells control the opening and closing of the stoma lah. Okay, palisade mesophyll and spongy mesophyll will be involved in the photosynthesis lah. They will have abundant chloroplast to absorb maximum sunlight for photosynthesis. So palisade mesophyll cell arranged vertically and closely packed lah, vertical columns like that lah. Okay, spongy mesophyll irregular shape and then they have air space between for gaseous exchange lah. Okay, vascular bundle, you have xylem and phloem. Remember, xylem is on top of the phloem. Okay, so xylem transport water and mineral salt from roots to the leaf. Walls of xylem are lignified and thickened to provide mechanical support and strength lah. Okay, that means they have lignin. Phloem transport the organic substances produced by the leaves lah, like all your starch, your glucose, sucrose, all these they will be transported by the phloem lah, and the sieve tube element. Okay. Okay, so main organ for gaseous exchange. Now everyone also knows lah, gaseous exchange of course is stomata lah. Okay, so the gas will enter and diffuse in and out of the cell through the stoma. Is it okay? Mechanism of stomata and opening and closing. Okay. So now you must know the mechanism of how the stoma open and close. Okay. If not mistaken, this one also maybe trial ask, is it? I forget already. Okay. So there are two factors that control the mechanism of stomata opening and closing. Okay, so you have two factors. One is the potassium ion, one is sucrose concentration. Okay, so one is because of potassium ion, one is sucrose concentration. Actually, the process is the same. It's just that the factor is different now. Okay, okay. Okay, so what causes the potassium ion is because the accumulation or elimination of potassium ion in the gut cell changes the solute potential. It will both of these, the process is the same thing, which is like because of the concentration, that's why low water potential, the water will diffuse in lah. It's the same mechanism only, just that the things are different. Like here is potassium ion, here is sucrose. Okay. So water is diffused in or out of the gut cell. Okay, determine turgid or flaccid lah. Sucrose concentration, remember, sucrose is produced by photosynthesis. So this one is about photosynthesis lah. So during daytime, there's photosynthesis. That's why we'll produce sucrose and then all that lah. Night, don't have photosynthesis, so sucrose concentration is lower lah. Okay, now, we look at the, this one is potassium ion first. Okay, so, at first, okay, opening how? Okay, opening. That means will become turgid lah. Okay, opening. Potassium ion. All the potassium ion go inside. Okay. The potassium ion all go inside for opening. Wait, did they explain? Hold on. Okay, so potassium ion go in. Okay, we look at potassium ion first. So potassium ion enter the cell through normal diffusion now. Okay, so you see, once the one inside the gut cell, the concentration increase. Remember, if the solute concentration increase, the water potential lower, right? Water potential lower. The neighboring cells, the one beside, water potential is higher. That's why the water will diffuse inside through osmosis lah. Okay, and because when you diffuse inside, it will become turgid lah. The gut cell become curved and turgid like that. That's why it causes the opening of the stoma. Okay, understand? Yeah. Now closing is the same thing. Okay, so closing of the gut cell is also the same thing. Okay, closing is because potassium ion come out. Potassium ion come out. Here the solute potential is low. Water potential is high. Outside water potential is low. So water diffuse out by osmosis. So it becomes flaccid and it will close. Okay. So sucrose concentration is the same thing. Okay. During daytime, you conduct photosynthesis during daytime. Here all this photosynthesis happening inside. So not sodium blood, sucrose will be high lah inside, sucrose inside. Okay, so water potential is lower lah. Outside water potential higher. Okay, that's why the water will diffuse in by osmosis. Okay, for potassium is the ion diffused in, but for sucrose it's not diffused in. It's because photosynthesis inside the gut cell. That's why sucrose concentration is high already. Okay, so the same thing during night time. The opposite thing happen lah during night time. Okay, no photosynthesis. Okay, so sucrose concentration very low. Okay, sucrose concentration very low like that. Okay, and then the water inside here, water potential very high, outside very low, so diffuse out by osmosis. Okay, do you understand the mechanism? So yeah, okay, this one actually you can also read your own basically like the control the opening and stoma control opening and closing for gaseous exchange and also to control the rate of transpiration. Okay, now main organ for transpiration. Now what is transpiration? Basically is I absorb the mineral salt and water from soil and then through the xylem from the bottom of the roots up to the leaf and then evaporation. This produces a cooling effect lah for the plants and also to obtain water lah for their growth. Okay, so okay. Now, there are 4 factors affecting transpiration, okay? Light intensity, temperature, relative air humidity, and air movement, okay? Light intensity. Higher light intensity, that means it will be hotter, right? Light intensity is slightly related to temperature lah. That means high light intensity will become hotter, more photosynthesis. That's why you need more water lah, more water for photosynthesis and also cool down the plant. Okay, to cool down the plant. That's why the rate of transpiration is high lah. But then you see, it will become constant. It will become constant once it reaches a level because of other factor. Maybe the air humidity, the temperature, and air movement. It will become the limiting factor lah. Okay, same for temperature because you see the temperature increase, kinetic energy of water molecules increases the rate of transpiration. That's why you need more water lah for transpiration. Air humidity dia opposite okay, is inversely proportional because remember air humidity, when humidity is high, that means like after raining humidity is high, very wet, that means very less sunlight. If very less sunlight, that means less photosynthesis and less transpiration. But if humidity is low, that means sunny day, very less water vapor, then your transpiration will be higher lah. Okay, so rate of transpiration becomes higher when the humidity is low lah. Okay, air movement also because if your air movement is higher, very fast, your air molecules, water molecules all removed from the plant. That's why you need more higher rate of transpiration lah. to produce the necessary water molecules to cool down the plant. Okay. Okay, so photometer, this one you can look through yourself. Okay, now, just now we have transpiration. Now we are going to photosynthesis. Okay, so brief history, this one also not important. You can read yourself. So how do they discover photosynthesis? Basically is Hellman, okay, Jan Baptista van Helmont. Basically what he do is he weigh the seedling 2.3 kg, 90 kg of dry soil. Plant was watered for 5 years, and then later on he weigh lah. Okay, he weigh that the plant is weight 77 kg. Okay, so his conclusion is the plant had grown by only being watered and not from the water of the soil. Okay, so that means that because of the water of him watering for 5 years, that's why the plant can only grow lah. Okay, for Joseph Priestley, okay, a rat was placed in an inverted glass jar. The rat died, but because when he placed the plant and the rat together in a glass jar, the plant produces oxygen during photosynthesis. That's why there's enough oxygen for the rat to live lah. Okay, so plants release oxygen. Okay, so adaptation, this one is also the same thing as just now lah. Okay, so transparent waxy cuticle and then stomata, palisade mesophyll packed with chloroplast, okay, to enable sunlight to absorb photosynthesis at maximum rate. Okay, spongy mesophyll also same thing, gaseous exchange, vascular bundle lah, xylem and fluor. Okay, now the new thing here is chloroplast structure. Okay, this is something new that you have not learned in lower farm. So chloroplast structure, when you are drawing in like a 2D diagram, okay, remember chloroplast is always double membrane bound organelle. And then you will have like this lah. You will have your thylakoid, okay, your grana, and then you will have your what is it called? You will have your lamella lah, okay. Now this is chloroplast. If you want to draw in detail, so this is how the structure is lah, okay, of chloroplast. Okay, you will have your thylakoid, okay, your stacks of thylakoid. So all these thylakoid stacked together, thylakoid, green color, contains the chlorophyll pigment here, contains chlorophyll pigment in the chloroplast. This is one thylakoid. When it's stacked together, it will form the granum. Granum, plural is called grana, okay? So singular is granum. And then you have multiple stacks of thylakoid, multiple stacks, not just one stack. You have multiple stacks of thylakoid, okay. And then another stack over here. And all this is connected by lamella. And then the region outside here with no thylakoid or granum is called the stroma. Okay, so light-dependent reaction occurs in the thylakoid or grana, and the light-independent reaction occurs in stroma lah. Okay, so last time we call light-dependent reaction as light reaction. Stroma, we call it as dark reaction, light-independent reaction. Okay, so look at the description. Thylakoid is a disc-shaped sac containing chlorophyll. Okay, this shape containing chlorophyll pigments inside. Okay, in thylakoid membrane, there are photosynthetic pigments that trap sunlight. Light-dependent reaction occurs in the thylakoid. It's called light-dependent because you need light. You need the sunlight for this reaction to occur in photosynthesis. Okay, for granum is a disc-shaped stack of thylakoids. Arrangement increases surface area lah. Okay, stroma is colorless fluid surrounding the granum. Site for light independent. Light independent means you don't need light lah to conduct the dark reaction. Okay, which produces glucose. Now, okay, chromatography is just to separate the chlorophyll inside lah, not important this one. Okay, now this one is the hard part lah, where students always struggle. Last time you learn photosynthesis, very easy only. You learn water plus carbon dioxide produces glucose and oxygen. But now at this level, it becomes more complex. You have to know the light-dependent reaction and light-independent already, okay? Okay, remember, light-dependent must occur first, okay? Light-dependent must happen first, baru you move to light-independent, okay? So first of all, light dependent, you must know it only involves the water. Okay, it only involves the water. Carbon dioxide pergi sini. Carbon dioxide akan pergi sini, water is here. Okay. So, first thing, what happens? Photosynthetic pigment on the surface of thylakoid absorb the light energy. Okay, light energy maksud lah. So, dalam chloroplast ni, okay, chloroplast, the light energy masuk lah, light. Okay. Okay, what happens next? Light energy excites electron in chlorophyll pigments to a higher level. Okay, so electron dalam chlorophyll, electron akan excited because of the light. What happens next? Actually, this part, some of the sequence, I think like they confuse you because I don't like that they, how to say, they, like you later you can see lah, the photolysis will occur down there and then they up here already mentioned then. So we just follow according to how I explain lah. Okay. So excited electron from the chlorophyll go through a series of electron carriers and then the energy is used to generate energy in the form of ATP lah. So all these electrons, okay, all these electrons excited already, they will go through an electron transport chain, okay, electron transport chain. Actually, to be more complex, there will be two photosystem, okay, there will be photosystem 2 and then there will be photosystem 1, but then that one is not in our syllabus, so we won't go too much to that. So it will go through a chain, electron transport chain, and then generate energy in the form of ATP lah. So there will be ATP. Okay. Okay. Eventually, this. Okay. Before I move on to that, we focus to another side. Okay. We focus to another side. So, like I said just now, ada water kan. So this water, okay, this water will be broken down into hydrogen ion. Okay. Water molecule break down into hydrogen ion and hydroxide ion. Okay. Now you see. So this process is called photolysis lah. Photolysis. Okay. What does it mean? Photo, that means involving light. You need the light. Okay. Lysis means you break down of water. OH. Okay. So this one. Okay, I think I better go to this, focus on here first. Okay, so you focus on OH first. This OH minus this hydroxide ion lose electrons. Okay, lose electrons. Okay, and then it will become the water again. Okay, so it will produce the water again. Now, where this electron go? This electron will go back up here to replace because this one is excited mah. Dalam chloroplast itu, this electron dia excited, dia go through electron transport chain and then later on we'll see where it goes lah. So this one will replace lah. The hydroxide ion lose the electron and then will go here to replace the one that is already lost lah, excited. Okay, and then you'll form water again. Okay, water habis, photolysis, and then like this lose electron already. Okay, habis. Water sampai sini. Now, kita pergi balik elektron. Pergi mana? Okay. Elektron. Okay. The H+. Tengok H+ ini. Elektron. Okay. And the H+, they will reach a site called NADP+. Okay. NADP+ combine with H+ and the electron to form NADPH. Okay, so the end result of this light-dependent reaction is NADPH. You need the NADPH for here, for light-independent reaction. Okay, can you understand? Light-dependent sampai sini. There will be two stages lah. Okay, like two different parts lah. The first part is the electron yang akan form NADPH pergi sini. The second one is the water, photolysis of water. Lepas tu dia akan macam ni. Dia akan lose electron to replace this one, and the hydroxide will become water. Hydrogen ion pergi dengan electron, combine dengan NADP+, form NADPH. Can you understand? Light dependent. Okay? Okay, okay. So once you know already light dependent, baru you pergi light independent. So you see what happens in light independent. 5 carbon organic compound fixes carbon dioxide gas to form 6 carbon organic compounds. So just now this one, okay, 5 carbon ini dia combine dengan carbon dioxide because another carbon here, that's why it forms 6 carbon lah. Okay, the second one, okay, NADPH and ATP. Okay, this one also ATP. So just now you already have NADPH, right? You have NADPH, you have ATP. So you reduce the organic compound to glucose monomer. So dia akan reduce 6 carbon ini jadi glucose monomer. Because remember, glucose is the monosaccharide, right? The basic unit of the starch. That's why dia akan reduce ini jadi glucose. Okay, and then glucose ini combine sama-sama a lot of glucose, okay, all this glucose condensed together to form starch lah. Okay, and then it's stored in the plant lah. This is how you form the starch lah. Okay, so that's all lah. Installed in the stroma of chloroplast. Habis. Okay, this is the light independent reaction. Now, for the word equation, okay, word equation, so you know lah, okay. Okay, we go to the what is it called, like the chemical symbol first. Okay, so water, okay, H2O, water and carbon dioxide. Okay, after you have sunlight and chlorophyll, your UV and chlorophyll, you will produce oxygen, glucose, C6H12O6, and also water again, H2O, because remember, here your hydroxide lose electron to form water again. So you actually have water, okay? So here, everything will be 6, and then here will be 12, and then here will be 6 lah. Understand how to balance it? Okay, 6, 6, 6, and then since here already got 6 water, here will be 12 lah. Okay, and then you have your glucose. This is the balanced chemical equation lah. Okay, can you understand process of photosynthesis? Can understand or not? So this is the similarity and differences. So light dependent reaction and light independent both catalyzed by enzyme lah, okay, and takes place in the chloroplast. But the difference is, you see, site of reaction for light dependent is thylakoid, site of the reaction of light independent is the stroma. Okay, involve photolysis of water. Remember, water is for light dependent, carbon dioxide fixation is for light independent. Okay, light independent doesn't use the ATP molecule. It will produce the ATP molecule because of the electron. Then the ATP molecule is used here in light independent. Okay, and then the reaction substance, okay, water. For this light dependent, you see, light dependent will be using this water to produce the water and oxygen. Okay, for light independent, carbon dioxide you use to produce glucose. Okay, and then yeah, I think that's it lah. Okay, for the similarity and differences. Remember, the light dependent dulu baru the light independent. So we move on. Okay, just now transpiration you have 4 factor. Now photosynthesis you have 3 factor. Okay, you have carbon dioxide concentration, light intensity, and temperature. Okay, so carbon dioxide concentration, of course, more carbon dioxide, that means higher rate of photosynthesis lah. You can produce more glucose. But you see, after a while, it becomes constant because of limiting factor, such as light intensity and temperature. Okay. Okay, light intensity also the same thing lah. Higher light intensity, that means more light energy, so more electrons become excited. Okay, but then it will become constant because of limiting factors such as temperature and carbon dioxide concentration. Okay, so temperature is the same thing, except you see it's a bit different because remember this is like your enzyme graph. The optimum temperature is 25 to 30. After that, it will reduce because the enzyme will become denatured. The enzyme denatured, so photosynthesis cannot occur already lah, beyond 40 degrees Celsius. Okay. All right. So this is effect of different light. You can read yourself lah, not too important. Yeah, this is also why we can see green color, because all the red light is absorbed by chlorophyll. Blue light is absorbed by carotenoid pigment. That's why we can only see the green lah. Okay. Okay. Now you have to know what is compensation point. So you have already learned about respiration. Now you are going to learn about photosynthesis. Okay, you already learned for respiration and photosynthesis. Respiration is like almost opposite of photosynthesis. Respiration, you uses oxygen and glucose to produce water and carbon dioxide and energy lah. Tapi photosynthesis, water and carbon dioxide to produce oxygen, glucose and water. So it is opposite lah. It complement each other. Okay. Now you see, in this graph, there is negative and positive, and then the zero is compensation point. Compensation point means level of light intensity when the rate of respiration equals the rate of photosynthesis. That means I respirate at this rate, photosynthesis also at the same rate. That's why my carbon dioxide will my oxygen produced by photosynthesis will be used by respiration, and then my carbon dioxide produced by respiration will be used for photosynthesis. That means it's continuously like that lah, the cycle. Okay, it's the same thing and same thing only. Okay, and then that's why there is no net gain or net loss in glucose, because it's the same rate only. That's why we call compensation point. Okay, but if photosynthesis rate is higher, then there will be a net gain in glucose. That means because you see, you produce more glucose than you use. Okay. If respiration rate is higher, there will be a net loss in glucose because you use more glucose than you produce. Okay. Okay. So comparison between the two. This one actually, Form 1 also you learn already. So similarity, both process takes place in living organism, involve uptake and release of gases. For photosynthesis, it's only in green plants and also organisms that contain chloroplast, chlorophyll, but respiration is for all living organism. Type of cell happens in the cell containing chlorophyll, respiration in all cells. Anabolism, remember anabolism, that means it's synthesizing the small molecule combined together to form a larger molecule, which is glucose. Okay, synthesis of glucose using carbon dioxide and water. Respiration, catabolism, that means breakdown. Big molecule break down into small molecule. So catabolism happens, breakdown of glucose to produce energy lah. Okay, chloroplast is the site for photosynthesis. Mitochondrion is the site for respiration. Reaction substance, carbon dioxide and water. Carbon dioxide and water is used for photosynthesis, whereas oxygen and glucose for respiration. Then the product also the same, byproduct, and then energy involvement. Okay, photosynthesis, you absorb light energy to produce chemical energy. Respiration, you use the chemical energy to produce the ATP energy and also heat energy. Okay, photosynthesis, you need light. Respiration, you don't need light. Okay, even though photosynthesis, there is one process, light independent, you don't need light. But before light independent can occur, you need the light dependent reaction to produce the NADPH and ATP. Okay, so that's all for the differences, and that's all for chapter 2. Any questions for chapter 2? If you understand, it's good lah. Honestly, if you understand, you don't have to really like follow word by word, the textbook word by word macam tu. You must have your important terms, then can already. Like here, maybe you can say light energy is absorbed by this chloroplast and then electron excited, all this lah. Your terms must be there. Keywords and terms must be there. But you don't have to follow 100% the word by word lah. Okay. So now we move to chapter 3. So chapter 3 is nutrition. Honestly, it's a very, very not to say complex, but a lot of things to memorize. Even I also cannot understand it. Because in humans, we have easier nutrition and also the explanation is easier. But for plants, there's so many elements that you have to memorize a lot, which I really don't encourage. Even I also don't like that chapter, okay? Yeah. micro and macronutrient. But luckily, because of friends lah, they give me tips already and how to memorize. So I will try my best to explain to you what are the tips and mnemonics for you to understand lah, the macronutrients and micronutrients. Okay, but don't go and memorize lah. Okay, because I don't think so it's worth it. Like you see in trial, only like objective only one question that chapter, and also subjective. Did it come out in subjective? I don't think so. So not really worth it for you to go and memorize those. Okay, nutrition. You have macronutrients and micronutrients. Okay, everyone knows macro means big lah. Okay, big needed in large quantity. Micro means small needed in small quantities. Okay, so how do you memorize macronutrients? Okay, macronutrients you have 1, 2, 3, 4, 5, 6, 7, 8, 9. Who you 9 elements. So the mnemonic they use for macronutrient is okay, perempuan. Okay, science. Okay, you know science in our jadual normally is SC or SN. Okay, SC lah in this case SC. Okay, so you see P here means phosphor. Is it phosphorus? Yeah, phosphorus. Okay, S here means sulfur. Right, sulfur. Okay, and then C here means carbon. Now, with carbon, automatically in your head, there will also be hydrogen and oxygen, okay? Because those are the building blocks of your, what is it called? Your carbon dioxide, your carbohydrate, and also your protein, your fats, all those. K here, potassium. Okay, M here, you see M and G, so magnesium. N here, you see, N here is nitrogen, okay? And then Ca over here is calcium. So how many? 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay. So this is your macronutrient lah. Perempuan sains Korea memang nak cash. Okay. Perempuan sains Korea memang nak cash. Understand? This is how you memorize the macronutrient. Okay. The function and deficiency is another story lah. That one also hard. But I actually another more an easier way to how to say, to understand this is not to memorize according to the nutrient, but to memorize according to their function. Understand? For example, formation of of chloroplast, for example, or chlorophyll. You only tengok which nutrient is responsible for that. Instead of going, okay, nitrogen buat apa, potassium buat apa. That one is very hard. Confirm you cannot remember one. Okay. So, in terms of function, the first one is formation of, okay, maybe you can say formation of protein or what lah, of amino acid, okay? Okay, formation of amino acid, you know protein, you need carbon, hydrogen, oxygen, nitrogen. So, all these elements, you know already lah, formation of amino acid. Okay? Okay. And then what else? One more thing I forget already. What is it? Okay. Your second function is formation of chlorophyll. Something like that lah. Formation of chlorophyll. Okay. For the chlorophyll, you just remember Maggie. Maggie is the noodle, right? The food, right? And your drink will be F and N. Okay. F and N. So Mg, F and N. Okay. So Maggie will be magnesium, F will be fluorine, and then your N will be nitrogen. So these three elements, nutrients, will be for the formation of chlorophyll lah. Okay, you memorize macam tu saja. Okay, what else Okay, phosphorus. Okay, phosphorus and enzyme. For enzyme will be phosphorus and sulfur. Okay, macam tu saja. So you just memorize 3 function and then the element, okay? First function, formation of amino acid, which is protein. You already know these 4 elements. Okay. Chlorophyll, Maggi, F and N. So these 3 elements. Enzyme, these 2 elements. Okay. Habis. Macam tu saja. Okay. And then you must know the deficiency lah. Normally deficiency you can explain in your own words when you see this lah. Okay, so if no amino acid, no protein, how? Stunted growth lah, because you cannot grow without protein, stunted growth. Okay, formation of chlorophyll. If no chlorophyll, how? There will be yellowing of leaves, which is called chlorosis lah. Okay, for this you also can say protein synthesis disrupted. Okay, enzyme, kalau tak ada enzyme, how? That means you cannot carry out the processes lah. Unhealthy formation of what lah? Stunted growth, all this lah. Okay, can you understand the macronutrient effect on leaf? That one, unfortunately, you really have to memorize yourself lah. But if let's say you know no magnesium, no fluorine, no nitrogen, cannot form chlorophyll, confirm is yellowing of leaf already, which is chlorosis lah. Purple, yellow, that one unfortunately you have to read yourself lah. Red, white, blue, all that lah. Okay, I cannot help unless you really found a good YouTube video that really can explain 100% in a simple term. Then please share lah. Okay, please share to me also. I need it lah. But I watched the video that my friends share with me is basically you just memorize these 3 function and then the respective element can ready. Okay, like I said, it's not worth it you go and memorize so much, because in exam it's not going to come out like one essay. Essay they cannot ask you all this one. This is so short. This is like structure only, the mostly. Okay, 1 or 2 marks only. Cannot go until essay like that one. Not worth your time to memorize. Okay. The way they present the information is really too compact. That's why students stress this chapter. Okay. Okay, so that's all for macronutrient. Micronutrient also, unfortunately, I cannot find any mnemonic so far. But if you have, please share lah, okay? So you have to know yourself lah, all these chlorine, iron, manganese, boron, zinc, copper, nickel, molybdenum lah. And then you have to read yourself lah, okay? Read yourself the function and effect of deficiency, okay? Unfortunately, I really cannot help lah, okay? Even I also struggle in this. Okay. Okay, and then you have to know what is NOP solution lah, okay? NPK. Okay, so this NPK is basically the most important thing in formation in the growth of plant lah, NPK fertilizer, okay? Nitrogen, phosphorus, and potassium. So NOP solution consists of all this lah, calcium nitrate, potassium nitrate, all this lah. Okay, NOP solution is to study the growth of the plant relating to the nutrients lah. Okay, and then this one normally won't come out in paper 3 lah. Okay, you see this one takes months, days and months to observe, so cannot lah. Okay. Okay, so that's the nightmare is over already. Is it important to memorize according to micro or macro? For NOP solution, you don't really need to memorize anything lah, because it's very rarely it comes out. If you ask me the function of nutrients, focus on macro more. Okay, all this macro you focus more compared to micro. All right. Okay, now the nightmare is over already. Now we focus on the second subtopic lah, which is easier. Okay, which is organ for water and mineral salts uptake. Of course, you know how to absorb the mineral salt and water in the soil is through the root hair cell lah. Okay, so root hair cell, this is just the structure lah and the function you have to know. Okay, so root collar, part of the root connected to the base of the plant stem. You have your root hair. That means to increase the surface area for the uptake of water and mineral salt, and then your root cap located at the tip of the root protects the root from damage when going through the soil. Okay, now the structure of root is like this. It's like a star shape, okay? So structure of root is almost similar to stem, but then it will be a star shape. Okay, it will be like this, a star or like cross shape like that lah. And then you have your 1, 2, 3, 4 like that. Okay, the main thing here is the xylem, the big one. The small one will be the phloem lah. Okay, so you draw a cross like that. like a plus sign, and then there will be 4 small circles beside. That one is your formation of your structure of your root lah. Okay, and then you will have your vascular cylinder, primary xylem, primary phloem, pericycle, endodermis. Okay, remember endodermis dulu outside, baru pericycle inside. Because remember, endodermis is protecting the inside, the vascular cylinder inside. That's why the pericycle must be inside. Okay, so epidermis, the function is to protect lah. Basically, is to protect the root lah. Okay, so thin cell wall, water permeable. Okay, elongate towards the lateral side from the outer wall. Root hair cell are not layered with cuticle to allow water absorption. Root hair cell have big vacuole to store water and mineral salts. Okay, now the cortex is the same thing as the stem lah. It's basically the layer between the endodermis and epidermis. Okay, located under the epidermal layer, thin cell wall facilitating water movement in roots. Cells are loosely arranged to facilitate gaseous exchange. Cortex consists of parenchyma. Okay, endodermis separating layer between cortex and vascular cylinder. Endodermal cells are closely arranged and one cell thick. Most of the endodermal cells have suberin or lignin thickening, or which form Casparian strip. Okay, so basically, okay, this one is later on more to transpiration. They will explain more. So basically, your pericycle or your endodermis, there will be lignin and suberin thickening. But then there will be a structure of layer called the Casparian strip lah. Because of this Casparian strip, so when you use your apoplast pathway, in the end, when you reach the endodermis, you have to retract back to simplast pathway. Okay, you cannot use apoplast to bypass the endodermis because of this Casparian strip. Later on, chapter 4, we will look more in detail for that. Okay, endodermis allows water and mineral salt absorbed from the soil to enter the vascular cylinder, but not air bubbles. Okay, vascular cylinder, you know lah, vascular bundle is xylem and phloem lah. Okay, so at the root core, xylem and phloem, pericycle involved in secondary growth. Xylem and phloem tissue arranged in star-shaped pattern, or the plus shape also can lah. Xylem tissue transport water and mineral salt, whereas phloem tissue transport organic substances such as sucrose and plant hormone. Okay, I think that's all lah. Oh wait, there's only one more small subtopic, which is diversity in plant nutrition. So basically, plants, you have 3 types, 3 special types lah. Okay, one is parasitic. That means like the plant, it will absorb the nutrient from the host cell, and then the host will eventually die lah. Okay, like this Rafflesia, it will absorb all the organic substances, mineral and water lah. Okay, and then you have the epiphytic plants. Epiphytic means it doesn't absorb the mineral from its host. It tumpang only, tumpang saja. That means you see like this bird's nest fern and orchid, it just grows on the host body, but it doesn't harm the host. Okay, dia tumpang the space saja to absorb more sunlight, all that lah. Okay, and then the third one is carnivorous. Carnivore means you eat the other organisms lah. That's why here you see the pitcher plant, the panther species, and also Venus flytrap will eat the insects lah. Okay, to obtain nitrogen in the insects. Okay, so yeah, these are all carnivorous plants lah. Okay, so yeah, that's all for chapter 3 lah, nutrition. Structure for what? Yeah, can draw structure for stem, root, and leaf. Can. It's easy what structure. Of course, your vascular bundle inside, and then you will have your layers lah. You have your endodermis, your epidermis, cortex, and pith lah. All this is easier. Or udicot and monocot lah. Depends lah. Maybe also. It's not too hard lah, monocot, udicot. Okay, but normally I think exam only ask you draw satu je kot. And normally it's udicot lah, not monocot. Okay, but as a biology student, you must know how to draw. It's not hard also, the structure of the plant. Humans lagi susah structure. Plants is not hard. Okay, can. No lah, you still have to be prepared and you have to know all lah. You cannot just, okay, I memorize udicot how ni, monocot I jangan. Yeah, at least you know lah. Let's say you know that, okay, in monocot, it's not arranged like that systematically. It will be randomly arranged like that, for example. Okay? Okay, so that's all for today's class. Please remember to fill in the Google form. Okay, so thank you for joining the class. If you have any question, feel free to ask me anytime. Okay, thank you and bye bye.